Researchers at the University of Vermont and Tufts University have teamed up to release a new and improved version of their living robots. We first told you about these so-called xenobots in January 2020. These microscopic robots are created from frog stem cells and can move on their own. Now, they broke down quickly within about a week. But I found out from UVM researchers this new version has a few upgrades. Joining me now is Sam Kriegman, a UVM postdoctoral assistant, roboticist, and co-author of this work. Thank you for being here.
Hi, Kat. Thanks for having me.
So let's start with what's new with Xenobots 2.0. What have you improved from your last version?
So the first paper, Xenobots 1.0, was a proof of principle that these things called xenobots exist, that computers can design them to behave in certain ways. And the second paper is about the steps we've taken towards making xenobots more useful tools.
So what did you do to make them more useful?
So in the first paper, it was kind of a painstaking process of creating an organism or a living robot from scratch. And this involved placing tiny heart cells within this robot, within these skin cells, to create this new kind of body shape. And this took a single microsurgeon a long time to do. So we created a way to make living robots much faster. And we created robots that instead of heart muscles, they use a different kind of motor so that they can move much faster. And this different kind of motor is called cilia. They're these tiny little hairs that naturally grow on a frog's skin to move around mucus, and they keep the frog nice and slimy and happy. But in the xenobots, these tiny little hairs are used like little oars on a rowboat. And so they allow the xenobots now to swim instead of moving with the heart muscle, which is kind of this very slow crawling.
So how did you overcome some of the obstacles to get those things to work?
So this advance is possible because of a rich body of work by developmental biologists that already knew how to produce cilia in different kinds of organisms. So we just repurposed this kind of new motor for our next generation of xenobots. And another thing that we did in addition to adding a new motor was adding a new kind of sensor for the xenobots. So now the xenobots have a little molecule inside of them so that they can behave, and if they experience a certain kind of light, when they come back home after behaving and we look at them under a particular kind of microscope, if they're glowing red, it means that they experienced this light source in their environment. So they can go out into the world and hopefully one day do something useful, or at least not interfere with whatever else is in the environment. They can come back home and they can report what they saw.
Briefly, what implications do these new features have?
So we're not quite sure if xenobots will ever be able to produce useful work. We hope that they will be, and we will work to try to make them as useful as possible. But they're already changing our conception of what a robot is, what an organism is, and what robots and organisms could be. And so that kind of thing, I think, is very important for science in general.
And so then, briefly, what's next for you?
What's next is, if we would like xenobots to be useful, in particular in medicine, we would like them to be made out of human cells instead of frog cells, and in particular out of a human patient's own cells, so that they can serve as a kind of immune system booster. They can go inside of our bodies and clean plaque from our arteries, or locate disease and targeted drug delivery. These kinds of things sound like science fiction, but they might not be that far off.
Very neat research. Always glad to hear about it. Sam Kriegman, thank you so much for taking the time to update us.
Thank you.